set of biochemistry slides where we're going to introduce proteins today. I know we've covered amines and amides in our previous lecture, and now we'll see the biological molecules that contain those functional groups. We did the same thing when we covered carbohydrates. We had just finished discussing the carboxylic acids and aldehydes and ketones and hydroxy groups from alcohols, and then we talked about carbohydrates, which contain those functional groups. So we'll do the same thing here. Our proteins are going to contain the amino and ultimately amide groups when we put them together into proteins. And so the first thing we need to ask is, what is an amino acid? And I want to think about the term amino acid not so much from a biochemistry standpoint at first, but just from a chemistry or organic chemistry standpoint. What does it mean to be an amino acid? Well, we've talked about hydroxy acids last time, or in the previous lectures, and it meant you had a hydroxy group or an OH group as a substituent on an acid or a carboxylic acid molecule. Here we mean the same thing, except it's an amino substituent on a carboxylic acid group. Right? So we have a carboxylic acid as our parent chain. On it exists an amino group. That's all amino acid means. And so you need two features to be an amino acid. You need a carboxylic acid and you need an amino group. And they may not necessarily be near each other. Right? So here's some examples in this figure. The top left figure is a very simple amino acid. You see the carboxyl group on the right, and then it has an amino group as a substituent. How do I know it's a substituent? Because this is the highest priority group. If this group had not been there, we could have named this as an amine, but it's not the highest priority. Sorry.
Nobody can see the screen. Like it's completely black for everyone on the Discord.
Let's try that again.
Yeah, we couldn't hear whatever you said after you said we're introducing proteins.
Okay, let's try it again. All right, how about now?
I can see it now.
Okay, excellent. All right, let's continue. Sorry about that. So we have a carboxyl group on the right, and we have an amino group on the left. And all this means is you have a parent chain with a carboxyl group and an amino group as a substituent. Okay, why is it a substituent and not the main chain? Because the carboxyl group has higher priority in your list. So you would simply name this as a 1, 2, 3, 4 carbon chain. This is not a carbon. It's a 4 carbon chain. So it's a what acid? What would you call that?
Carboxylic acid.
Right, but what's the parent name? 4 carbons.
Butanoic acid.
Butanoic acid, correct. And the substituent would be called?
Amino.
Amino, and its location?
4.
Right, so this would simply be a 4 amino butanoic acid. Right, so you could easily name that. We did this before. It's rather simple. Right, locate your highest priority thing and name the substituents. The second one is no different. It's just a little more complicated of a substituents. So here we have a carboxyl group again. We have an amino group. We have a parent chain or main chain that's 1, 2, 3 carbons long. This is not part of that chain because you can't go in and out of the carbon ring. So 1, 2, 3. This carbon's in the ring. So just 3 carbons long. And it's a propanoic acid. There's an amino group on 2. And there's a cyclopentyl group on 3. Right, everybody understand that part? Amino group on 2 and cyclopentyl group on 3. Amino would come first alphabetically, so you'd list it as a 2 amino, 3 cyclopentyl propanoic acid. Okay, so these aren't very complicated. It's all the rules you've seen before. You're just now naming it as an amino substituent on a carboxylic acid. That's all amino acid means. This one on the
You're frozen again if you're talking.
Yes, I don't know what's going on, why it's not keeping up.
Yeah, you keep cutting in and out.
Okay. Okay. So it may have been an internet connection since it's happening on both platforms. Right, sorry about that. Can you guys hear me now?
Yes, but it's still cutting out a bit.
Okay, so it's probably the internet connection, and I have no control over that.
Okay.
So I'm sorry. If it's doing it on both platforms, that's very likely.
You are recording though, right?
So the molecule on the right is also an amino acid because it's got a carboxyl group, and it's got two amino groups, in fact, right? But the one on the right, I'm not going to ask you to name. It's beyond the scope of what we've done so far in the class. I won't ask you to give me its IUPAC name or anything like that. But I want you to recognize it as an amino carboxylic acid, right? It's an amino acid. It has those functional groups. Okay. Now, what you've probably heard before, the term amino acid or amino acids, referring to a set of common amino acids. There are 20 naturally occurring common amino acids that we're going to discuss, and they're all what we call alpha amino acids. And what that means is the amino group, like this one over here, right, is not on this position, but it's on the alpha position, right, or carbon number 2. Remember, number 2 is the alpha position. So when we say the common 20 amino acids used in proteins, they're all alpha amino acids, meaning the amino group, much like this one in the center, is positioned on carbon number 2. Right? And then what's attached past carbon number 2 varies quite a bit. Right? So this thing varies what's attached out here, and that's our different 20 amino acids. None of these are among those 20, but this is the closest one to them since it's also an alpha amino acid.
Excuse me?
Yeah.
Sorry to interrupt, but nobody can see your PowerPoint anymore.
Right, so I don't know why. Maybe an internet connection, but I can't control that if the internet keeps cutting in and out.
So should we just pull up the PowerPoint on our own?
You could. I'll try to refresh it if you let me know, but I can't stop it from doing that.
Can you just tell us what slide you're on, and then we'll just follow?
Right, I'm on slide number 2 at the moment.
Okay, thank you.
We can see now.
Yeah, so if it cuts out again, we'll just remind me. I'll try to fix it, but I apologize. I can't control the internet connection. But what we're talking about now is the 20 common amino acids down here, where you have a carboxyl group, of course, like all of these do, and they have an amino group, just like all of these do, but it's on the second carbon, carbon number 2. So like this one in the center, all of these are alpha amino acids, and they vary by what's attached to carbon number 2. Right, so it could be a longer carbon chain for most of our amino acids, and for one of them, that's the end of the chain. It's just a pair of hydrogens here. So this side chain at the bottom, what we call the R chain or R group, can vary among the different 20 amino acids. Okay, so here's a picture of a typical one of our 20 amino acids, where again we have our carboxyl group on number 1. You have your amino group attached at number 2. This is the alpha carbon, or number 2 carbon. And then beyond that, you have the rest of the chain, whatever it might be. Right? There's also going to be a hydrogen here, because this carbon needs 4 bonds, of course. So this carbon will be chiral. Right? There'll be 4 separate things attached, assuming R is not another hydrogen. And that'll be true for 19 of our 20 amino acids. So this R group is kind of whatever it might be for our various amino acids, but they all have this in common. Right, so this base is always the same, or this common precursor, the first 2 carbons are always the same for our 20 amino acids. And what's attached at R is what varies. Okay? This is how you've been drawing it and seeing it for a lot of our molecules. There's no charges. Everybody has the appropriate number of bonds, right? Nitrogen making 3, carbon making 4, oxygen making 2, hydrogens making 1, and so forth. We've been seeing it that way the whole semester. But realistically, this is not how it appears in solution or in an aqueous environment, right? Because this carboxyl group is a carboxylic acid. It is a weak acid. And this amino group is a weak base. So it's very likely at a neutral pH that this will be deprotonated. It's lost its proton here. You see the proton is left, not the whole hydrogen, just the proton. It left the electron behind. So this has an extra electron, so it's a negative charge with 3 lone pairs. And very likely this amino group, this nitrogen, acts as a base, and its lone pairs can make a bond to a proton, generating the positive charge. It may not be the exact proton from here, but you can think of it as that one. And so it has all the same parts, it's just been rearranged. The extra proton from here is now over here. That's why it says H3 instead of H2. Right? And this is called the Zwitterionic form. Right? So it has both charges in the same molecule. Overall, it's still neutral, but it does have a separation of charges now. Right? And of course, since chemistry was mainly started by German-speaking scientists, They get to name everything, and Germans have great words for everything. And this is called Zwitterionic because it's just named two ions. If you speak German, you'll understand the prefix, right? So zwei means two in German, so it's a two-ion molecule, so it's Zwitterionic. And this one would be non-Zwitterionic. It doesn't have the pair of ions. Right? And you can think of it in this manner, where the proton from the carboxyl group has been transferred to the amino group, giving it this form in solution. Now, which of these two do you think would be more soluble in water? The non-Zwitterionic form or the Zwitterionic form?
Zwitterionic?
Yes,
Zwitterionic.
the Zwitterionic. Why?
Because it's a positive and negative, it's ionic.
Right, because it's an ion. Exactly. It has all the exact same parts. It's just the charges have been created by moving that proton around. And the more full charges you have, the more soluble in a polar solvent like water you will be. And so that's the case. So the Zwitterionic form makes these amino acids much more soluble than they would be if they existed in this form. And many times you'll see them drawn this way. And I'm okay with you drawing it that way while you're learning it, but know that they'll never exist like this in reality. There's no way it'll have this protonated and this deprotonated at any particular pH. It just won't happen. So just know that this form exists, and that's how it exists in reality, making it much more soluble than you think it would be if you tried to draw it like this. And that's shown at the bottom as well if we change the pH of the solution. So if you're around a neutral pH, and I don't mean exactly 7, I mean anywhere from like 5 all the way to 8, anywhere in that range, right? It's going to look like this, right? Because the amino group will be protonated and the carboxyl group deprotonated. And the reason I gave that range is because it's between the pKa's of those two groups. I'm not going to ask you to memorize those, but know that at a certain pH, this thing will favor protonation over deprotonation, and at a different favored pH, this one favors protonation over deprotonation. So if I make it very, very acidic, right, very low pH over here, right, lots of protons being added, then they'll both be protonated, right? And that's a common way of seeing this at very low pH, like pH 1 or 2. If I make it very, very basic, somewhere around pH 10, 11, 12, something like that, it'll be both deprotonated, right? This will be NH2 and this will be COO minus, right? And anywhere in between, it'll have this Zwitterionic form. So it's more soluble in this form than it is at extremes of pH. And here's our 20 common amino acids that I want you to learn. And I've put this slide up for you before on previous lectures, so you can start looking at them. And if you notice, all of their bottom parts are identical, with the exception of proline here, right? But they're all identical, where they have this carboxyl group and an amino group on number 2. There's generally a number 3 carbon in every case except the first one. I told you there would be one where they didn't have that carbon, but that carbon's present in all the other 19, and some are quite long in their carbon chains, and they have various functional groups attached at the ends. Right? And some are very, very simple. They're just hydrocarbon chains, right? We call those hydrophobic ones. Occasionally, there's one that wraps back around to its nitrogen. That's proline. It's an unusual one. This one has a sulfur in the middle of the chain. That's methionine. Right? And then some are alcohols, like these up here. They've got OH groups up there. Right? Here's another one that's got a big phenolate group on it. It's also an alcohol. And here's some more greasy ones with just benzene rings or an indole ring. And then we have some acids on this side. So this carboxyl group up here looks like this carboxyl group, right? As does this one, just on a longer chain. We have some where the carboxyl group is now an amide. Right? So we get a pair of those that correspond to these. Then we have our 3 basic ones with amino groups on the end down here. Right? So it seems like a lot to learn. There are 20 different ones. But if you break them up into smaller categories, it's easier to learn. So here on the left, I broke them down into their smaller categories. If you add up all these letters here, yes, there's more than 20 present because some of them appear in multiple categories. For example, if I ask you which ones are alcohols, right? There's only 3. There's serine, because of the OH, threonine with an OH, and tyrosine with an OH. These are carboxyl groups, so they're not alcohols. So these 3 are alcohols. There's only 3. So it's easy to remember them in small groups like that. But if I also ask which ones are aromatic, which one contain benzene rings, well, that's this group here. Phenylalanine, tyrosine, and tryptophan all have a benzene ring in it. But you see tyrosine appeared in both of those subcategories, and that's entirely possible. See, I wrote it here. Aromatic and alcohols, they both have tyrosine in them. But if you remember them in small groups, it's so much simpler than trying to memorize a list of 20 things. Right? When you were memorizing your phone number, you probably memorized it, if you guys even memorize it anymore with your phones. But you memorized it in small groups of numbers, not 7 numbers, but in groups of 3 or 4 or 2 or 5 numbers. Right? Depends on your nationality with your phones. But if you're memorizing these things, try to do them in small groups. The largest group is your hydrophobics. It's a really large group, but they're easy to recognize because they're generally just hydrocarbons, right? So not this one, of course, but glycines, alanine, valine, leucine, isoleucine. All these that are just not very interactive with anything but other hydrocarbons, right? So it's fairly easy to know that huge list. And then you've got 2 acids, because they're carboxylic acids on this side. Yes, these also have carboxyl groups. I agree, they all have that. But this one has an additional one, so we're going to call these the acid ones. There's 3 basic ones with these amino groups, and there's 2 amides, which are derivatives of the acids. And that takes care of about half of your molecules right there. There are 2 with sulfur. There's one with a sulfhydryl group, cysteine, and there's one with a methio group, methionine, methylthio. That's why it's called that. Right? So some of them are rather simple to memorize. For example, phenylalanine. Sounds like a long name, but it's a phenyl group attached to what would be otherwise an alanine. And so it's a very easy name to remember. If you know alanine, put a phenyl group on it and you got phenylalanine. Right? So some of the names don't make sense, I agree, because they don't resemble anything else. But a pair of them, I'll let you figure this one out. Of all 20, only 2 are isomers of each other. And I'll let you figure out which 2 they are because it's pretty easy to do. But only 2 are. 19 of these 20 contain at least 1 chiral center. This number 2 carbon is chiral in every case except the first one. And you probably recognize why right away because it's got 2 of the same thing on it. None of the others have 2 hydrogens there or 2 of anything there that are similar. So they're all chiral at that position except glycine. It's the achiral one.
I have a quick question. You just answered it. I saw on the practice test achiral. That just means like not chiral.
Correct.
Right?
Like acyclic means not cyclic. Right. The A prefix means a negative. Right. Good. So let's move on to this figure, which I found online somewhere many, many years ago. I don't remember where I found it, but be careful when you pull up drawings online, right? Because there are often mistakes in them, and you will find lots of them looking online. The mistake is sometimes not intentional, of course, because they're trying to be quick about drawing this, or they're being lazy, or they just don't know the answer. So be careful when you're looking these up that someone's not made a mistake. So in this figure, there's 2 mistakes. Right? It's a nice figure in that they put all the charges correct, and they highlighted all the R groups for you in colors and grouped them and everything, but there are mistakes. Look at this proline. This is clearly from laziness. Right? So if you look back at proline real quick, you notice how it wraps around to its own nitrogen. The R group is just 3 carbons, but it bonds back to its own nitrogen. It's the only one that does that. So they were being quite lazy here and just copying this NH3 plus over and over and over and left it here. This should not be NH3 plus. That nitrogen has 5 bonds now. Right? It should say NH2 here, not 3. So I was just being lazy. They didn't fix this figure. You notice this one shows both hydrogens individually, not all 3 like these. And then the other mistake in this drawing is this histidine down here. They also got in a habit of just showing this first group, right? And then they looked over at histidine and saw, oh, it's just this imidazole ring out here. But no, there is a carbon here, right? This beta carbon does exist, and they did not draw it, right? So it's missing the beta carbon here. So be careful when you look at drawings online or on your phone apps to help you memorize these things. There's lots of apps that you can get for free to help you memorize your amino acids, but maybe look at the structures once over to make sure there's no mistakes and you memorize a mistake. Right, so let's practice a little bit. So here's 4 examples of molecules, and I don't want you to tell me which amino acid it is yet. I know you're just learning them, but I want you to recognize yes or no, is it an alpha amino acid? Okay, so we'll go through 2 questions for each one. First, is it an amino acid? And then, is it an alpha amino acid if you answer yes to the first question? So looking at A, is it an amino acid, period? Does it have an amino group, and does it have a carboxyl group?
Yes.
Yes, exactly. There's the amino group right here, and there's the carboxyl group. They're on the same chain, so that is an amino acid. More specifically, is it an alpha amino acid?
Yes.
Yes, because the amino group's on the alpha or number 2 carbon. I agree. And in particular, this is one of your 20 common alpha amino acids. It happens to be threonine, right? You may not have learned that yet, and you're in the process of memorizing them, but you have to get used to seeing them different ways. They're not going to always be drawn this way, right? So if you're looking at threonine, here it is. You have to memorize this R group and see that on the alpha and beta carbon, there's an OH and a methyl, and you can see that here. On the beta carbon, right here, alpha, beta carbon, there's a hydroxy and a methyl group. Right, so get used to looking at them all different ways. How about choice B? Is it is it an amino acid at all?
No,
Why not?
it doesn't have the OH.
It doesn't have a carboxyl group, right? It's got the carbonyl, right, and it's got an amino group, but it doesn't have this carboxyl arrangement. So this is not an amino acid. What would you call it?
A ketone?
Right, it would be an amino ketone. Exactly. But we could be a little more specific. There is an amino group and a ketone, but since they're on the same carbon, what would you call that? We talked about this one last time. Say again?
Primary?
A hemiketal?
No, it's not a
It's an
hemi.
amide.
It's an amide. It's an amide, exactly. So it's like a carboxylic acid, like this, but instead of an oxygen here, there's a nitrogen. And it would be primary, like she said, because there's only one thing on the nitrogen other than carbon. I agree. So it's a primary amide. So it's not an amino acid at all. Okay. How about choice C? Is it an amino acid at all?
No.
No.
No, it's missing what?
The carboxyl.
It's missing the carboxyl group. So what would you name this as? What's the highest priority thing? What would you give it its name?
Alcohol.
It's an alcohol, right? So alcohol is of slightly higher priority than the amine, so we'll name it as an alcohol with an amino group, exactly. And then the last one over here, is it an amino acid at all?
No,
Why not?
Yes.
because it's not attached to the carbon.
So it is an amino acid. It has a carboxyl group right here, and it's got an amino group, right? And then my question after that, is it an alpha amino acid?
Yes.
Yes, it's on the alpha carbon, just like this one was. So the amino group is on carbon number 2. That's what's required to be an alpha amino acid, right? And so this one's amino acid because it has the amino group and the carboxyl group, and more specifically, it's an alpha amino acid because it is on number 2, right? And it is one of our 20 common amino acids. This one happens to be what, if anybody knows, based on this R group? You can look back on the previous slide. Right, it's just a long hydrocarbon chain that splits at the end. Right, so it's counting from the alpha carbon, it's 1, 2, and then it splits. What amino acid would that be?
Leucine?
It's leucine, exactly. So it's the 2 carbons and then it splits, right? So from the alpha carbon, 1, 2, and it splits. So this is leucine. Right? It's okay, you're still learning them, but I want you to recognize what is and is not an alpha amino acid first. And then we'll apply it to our 20 common ones after that. Okay? So let's talk about putting some of these alpha amino acids, or amino acids in general, together. You've done this in your lab exercise, where you took some carboxylic acids, like this one, and some amines, like this one, mixed them together, and you know a carboxylic acid plus an amine has a dehydration reaction. You get water loss, and you connect them and make the amide. Right? In this case, this one would be a, what kind of amide? Primary, secondary, or tertiary?
Tertiary?
Are there 3 non-carbon things on the hydrogen? Or sorry, on the nitrogen?
Secondary.
Secondary, because there are two non-hydrogen things on the nitrogen. Exactly, one hydrogen left. Right, so an example of that, and the one you did in lab, I think you guys did this in lab this semester. They've been doing it for a while. They take adipic acid, which is a dicarboxylic acid, right? And in hexamethylene diamine, right? Which means each of these can react with the other one, and it can keep growing into a chain. If I take another adipic acid and place it here, then this amino group could react with the second carboxyl group of the other adipic acid. And they keep alternating back and forth to make a really long chain. And this is how we make nylon, right? And it's called nylon 6,6 because there are 6 carbons here and 6 carbons here, right? And different style nylons, could be a nylon 4,3, for example, could have 4 carbons here and 3 carbons here, right? This one happens to be 6,6. And you notice it keeps making these amide bonds between this group and this group, between this amino group and the next carboxyl group, between the carboxyl group and the next amino group, and so on. And you make this really long chain. And some of you may have gotten it several meters long in lab. It's fairly fragile while it's still wet because these bonds can be broken by water. But if you let it dry, it gets quite strong. Right, so you may have done that in lab. We're going to do the same thing when we make proteins here, except we're not going to do it where it's alternating molecules. We'll use the same molecule with a different R group over and over and over. In this case, instead of having a carboxyl group on one molecule and an amino on the other, we have them on the same molecule, right? It's an amino acid. We have a carboxyl group and an amino group on the same molecule. And in a separate amino acid, I have another carboxyl group with an amino group. So it's not alternating molecules here, it's just we use different ones and they connect to each other. The R groups do not get involved in this reaction, they're just along for the ride. So you can have the carboxyl group from this amino acid and the amino group from the next one interact and form an amide bond. Of course, we lost water in this process. Right, this amide bond we're creating between the carboxyl of one and the amino of the next is often called the peptide bond. Right, and it's only called a peptide bond if it's between two amino acids. Right, these back here were not called peptide bonds. They weren't between amino acids. It's just an amide here. Chemically, this is still an amide, but it's between amino acids, so we often call it a peptide bond because it's the backbone of a peptide or a polypeptide or a protein. Okay? So if we make this bond, it has some interesting characteristics. Right? This amide bond has something we call resonance structures. So it can exist like you see it here, which is redrawn right here showing you all the lone pairs. Right? The carbonyl with its 2 lone pairs on the oxygen, the nitrogen with its 3 bonds and 1 lone pair, but can also exist in this form. Right? So the double bond here migrates between the carbon and nitrogen. Right? So this double bond, it's actually the lone pair here. It goes between the carbon and nitrogen. And this double bond, one of its pair of electrons goes up on the oxygen to make a 3rd lone pair. Right? So it's the shifting of electrons around. It's always happened here. Of course, this should have a negative charge up here they didn't draw. That goes with this, and a positive charge here. But it can shift back and forth. This one's clearly a little more stable than this one, but both do exist. And then when you say both of them exist, they can switch back and forth all the time, very rapidly, and we call those resonance forms. This was very similar to when you were drawing your Lewis structures. When you were drawing them and you got multiple Lewis structures as answers, and they were all valid answers, that made your molecule more stable. Right? The more you could draw, the more stable resonance structures you can draw, the more stable your molecule was. And that's what makes this peptide bond rather stable, because it has this double bond shift of a resonance structure. But that gives us a problem. So I'm looking at this molecule over here. Let me ask you a couple questions, or even this one, it's the same thing. Can this bond between the carbon and the R group rotate? Right? It's a single bond, so is this bond able to rotate?
Yes.
Yes, single bonds can rotate, right? The carbon-hydrogen single bond could rotate, but that's kind of a silly question. It's just one atom there. Rotating it does nothing, right? But this is a bigger group. It could be a huge R group, and rotating it could have some consequences. Can this bond rotate between the carbon, the alpha carbon, and the carbonyl carbon? So between carbons 1 and 2 here. Can this rotate, this single bond?
Yes.
Sure, single bonds can rotate. What do you know about double bonds, however? can double
They
bond.
don't rotate.
So,
They cannot rotate, right. So that we either said they were cis or trans, right? They can't rotate. So what about this bond between the carbon and the nitrogen? Can it rotate?
yes.
You would think yes, right? Because it's a single bond. But in reality, it's not always a single bond. Remember, it has this resonance structure. So it has what we call double bond characteristics, right? So this has a double bond character that it's not always a single bond, it's not always a double bond, but if you have a double bond or a partial double bond characteristic, then you behave like double bonds do and you can't rotate. So in this case, this bond, while it may look like a single bond, cannot rotate because it has these double bond characteristics. Right? So our peptide bonds do not rotate, which puts a constraint on the forms in which our protein chain can fold. Right? So here's another drawing showing the similar reaction. We're going to lose this OH and one of these nitrogens. They chose this one, and these two disappear to make a water molecule, leaving the carbon bonded to the nitrogen in our new peptide bond. And this bond has this double bond or partial double bond characteristic, so it cannot rotate. So you can treat it as if this thing were trans, right, the whole time. It can't rotate, so this can flip over and be up here. Right? It'll stay in this conformation. Right? Everybody understand that? I know it looks like a single bond, but because it has this resonance, it cannot rotate. It has partial bond characteristics. Okay, so let's start putting a few of these together. So here's a cartoon of a very small peptide called oxytocin, right, which is used in the body as a signaling a messenger hormone, right? And here's a more drawn out chemical structure of the same molecule, right? So what it's showing on the left is all the amino acids that are put together in order, right? And then on the right are all those amino acids drawn out in their chemical structures. Right? And you notice as this goes through, if you start up here at cysteine, right, here's the first one, and we go all the way down to the bottom through this S-shaped thing, right, we have cysteine bonded to tyrosine, bonded to isoleucine, to glutamine, and so forth down all the way to glycine. The cysteine here is actually connected to another cysteine, right? It's not from the beginning, but this is something that only cysteines can do. Remember, cysteine has that sulfhydryl group. Go back and look at it. It's an SH functional group on it. And SH groups can form what are called disulfide bonds. So you get one S bound to the other S. They lose a pair of hydrogens between them. And that's shown right here. Right? So this is the beginning of the molecule. There's the amino group. Right? I want you to be able to walk through this and recognize what's what. This looks complicated, but it's just a bunch of similar parts put together. Right? So you have the amino group, the alpha carbon, and the carbonyl carbon. Right? That's our direction we're going. So amino group, alpha carbon, carbonyl carbon. Here's the next amino group, the one from the tyrosine. Right, so this is the peptide bond. Amino group, alpha carbon, carbonyl carbon. Here's the R group of tyrosine sticking off. Right, this was the R group of the cysteine. If we keep going, here's another peptide bond between the isoleucine and the tyrosine. Right, amino group, alpha carbon, carbonyl. One more time. We keep following that N alpha carbon carbonyl pattern, NCC. NCC, NCC, all the way through. Right? So if you can pick any nitrogen in this molecule in the main chain, let's pick this one here. How do I know which way it's going? Left or right? Well, if I try going to the right, it's N carbonyl carbon. That's the wrong direction. It should be N alpha carbon carbonyl. N alpha carbon carbonyl. I know it's going this way. So you can pick any nitrogen in the backbone and figure out which way it's going. So in this case, I can start looking at NCC, 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 all the way through until I get to the very end where I have NC carboxyl group. It ends in a carboxyl group because there's no amino acid after the glycine. It began with an amino group. So we call the beginning of it the amino terminus, and at the very end of this peptide, we call it the carboxy terminus. So it's from the beginning to the end. That's the direction we orient ourselves for these amino acids. Okay? And it's labeled as you go through. This is a sulfhydryl group of cysteine. This is the R group of tyrosine, the big benzene ring with the hydroxy. Right? Here's our isoleucine group. Right? And then over here a glutamine, an asparagine, another cysteine, but this one is also bonded to the first cysteine. It's making a disulfide bond. Cysteine's the only only one that can do that. So it's actually forcing this into a ring, as you see over here. And then we end with a proline. Remember, the proline's R group wraps back around to its nitrogen, like that. Right? So this one has no hydrogen on it, right? Like the other ones still have their one hydrogen all the way through, right? But this one won't. Right? And then we have a leucine and then a glycine. Glycine's simple because it's just two hydrogens. They're not shown. We don't have to show hydrogens on carbons, but we must show them on everything else. Okay? So if we look at oxytocin here, it's rather simple. Let's change it up and look at a different one. This one's called vasopressin. It's very, very similar to oxytocin. So if I look at oxytocin sequence, there's a cysteine, tyrosine, isoleucine, glutamine, asparagine, cysteine, proline, leucine, glycine, just like we saw on the previous slide. If I look at vasopressin, it's got a few changes. Number 3 is not isoleucine, it's phenylalanine. So one hydrophobic to another, not a huge change. And leucine to arginine at position 8. So leucine is hydrophobic again. Arginine is one of your basic amino acids, so that's quite a change, right? And that gives you a different effect. So it's called arginine vasopressin because that's the main change we're interested in. In porcine, in pigs, there's also a lysine vasopressin. So instead of changing leucine to arginine, leucine is altered to lysine, another basic amino acid, but very, very similar to arginine. So it's almost the same as the human vasopressin, but this one's slightly different in pigs. And then there's one in fish, and it's also found in the human fetus, called vasotocin. It's kind of like we threw the words together and blended them up, because it has an isoleucine at position 3, like oxytocin, but it has an arginine at position 8, like vasopressin. So they call it vasotocin. Right? So it's similar, kind of a blend of the two. The disulfide bond is still present between numbers 1 and 6 in every case. So these are examples of showing how the chain can change amino acid components because there's a mutation in the DNA that codes for it, of course, or it's how it's supposed to be in that different organism, or a time of development, like in the fetus versus an adult. But it shows you that they're still put together the same way, from amino side to the carboxy side. Right? So how do we know they're put together this way? What determines the order of the amino acids? Well, it's determined by the DNA itself. And in that DNA is a combination of bases. It's a codon, 3 at a time. It's red. And we'll talk about this in a couple lectures when we do transcription and translation. But it's a series of codons that describe what amino acid should be assembled next. And when we were discovering this or learning about this in the 1950s, one of the first things done was to take a protein and start chopping it up piece by piece. So cut off one of the amino acids. So take a piece like this is the primary structure of the protein, the sequence, and start chopping them off one at a time. Right? And Fred Sanger, the biochemist from England, did this, and he won the Nobel Prize a few years later for the method he used to cut this up to figure out what amino acid was in what order. So he cut it off literally one at a time to figure out what was the next amino acid in the sequence. You could take the protein and degrade it completely with some very strong chemical agents, and you could tell there were X number of prolines in it, and Y number of glycines, and Z number of methionines, and so forth, but you didn't know what order they were in. He discovered how to take it apart one piece at a time to tell what individual amino acid was next. And by doing that, he was called sequencing. He figured out what the sequence was, or the ordered sequence of amino acids. From the amino terminus, we often call N terminus, to the carboxy terminus, or the C terminus. So we label it from the N to the C termini. Right? And then also Sanger, several decades later, in the 1980s, won a second Nobel Prize in chemistry. In fact, he's the only person to ever win 2 Nobel Prizes in the same field. Several people have won Nobel Prizes in different fields, most likely the Peace Prize and such. But from a scientific standpoint, nobody's ever won 2 Nobel Prizes in the same scientific field except Fred Sanger. So he got one for doing a similar process later on nucleic acids, taking them apart one nucleotide at a time to sequence those. So he's the only person ever to get both Nobel Prizes in the same field. Right, so speaking of primary structure, what did he mean by that? It's the ordered list of amino acids. I just want the order. I don't want an inventory of parts. I don't want there's 4 of this amino acid and 7 of this one and 12 of that one. That's just a recipe list. It doesn't tell me the order of them. So to have primary structure, I need to know the order. For example, here, it's a glycine, then a proline, then a threonine, then a glycine, and so forth down the line. I don't just give you a recipe. I give you what order they're in. That's called primary structure. And what holds primary structure together is simply the peptide bonds or the amide bonds in the backbone. That's it. And then we start moving up to what we call secondary structure. So this is where the protein starts folding on itself. Right? So it has interactions amongst the other parts of the protein. And these are mainly held together by interactions among the backbone. And if you remember in the backbone, we have a carbonyl every third atom, right? So it's an amino group, an alpha carbon, carbonyl. Amino group, alpha carbon, carbonyl. Right? So here's an example of that. Amino group, alpha carbon, carbonyl. And as we go around, amino group, alpha carbon, carbonyl. Amino group, alpha carbon, carbonyl. All the way around, down the chain. And those things, the amino groups, have a hydrogen on them. And the carbonyls, of course, have an oxygen on it with some lone pairs. Those can interact in something called hydrogen bonding, where you get a lone pair of one electronegative atom, an O, N, or F, and there's no F's in here, so it's always O or N. And then you get a hydrogen on another electronegative atom. In this case, we have a hydrogen on a nitrogen. So they're going to interact with what's called a hydrogen bond. We've talked about these before. And they can interact with themselves in several ways. Here's a hydrogen bond here. Carbonyl's oxygen to the amino group's nitrogen. Right? So it's making a hydrogen bond. It's not a covalent bond. It's just an interaction, an association. It's not nearly as strong as a covalent bond, but it is in large numbers strong enough to hold this thing into this shape. So this is called an alpha helix. Right? So every amino acid is hydrogen bonding to the amino acid 4 amino acids down the chain. So 4 down the chain will be the next partner. So if this is carbon, the carbonyl of amino acid number, let's say 16 in this chain, and if I move down the chain to 4 more, right, move down to number 20, it's going to be interacting with the amino group of number 20. Right, so it's always 4 down the chain. And if you look at a typical alpha helix, it's at minimum 5 amino acids long, because you need at least the 5th one down the chain, number 1, hydrogen bonding to number 5, that's 4 down the chain, to form the alpha helix in the first place. But they're typically longer than that. They're generally a little longer, about 11 amino acids. They can be as long as 20 or 30 amino acids, if you're going to cross the membrane, for instance. So they twist on themselves in this helical manner. It's often drawn as a winding pattern here. It's always a right-handed twist. If you ever see a left-handed one, it was drawn as a mirror image incorrectly. Right? But it's a right-handed amino acid or alpha helix twist. And if I went along the chain and asked where are all the R groups, because they're clearly not involved in this bonding, they're all sticking away from this chain radially. Right? So here's a way of looking at it. There's kind of down the barrel. There's no hole here because it fills up all the space. But if you were to look at it end on, right, all the R groups are facing outward from this thing. Right? And if you were to go along in numeric order, from the first to the second to the third, here they're in the 300s counting along this thing. It doesn't mean it's 300 amino acids long. The protein is that long, and this section of it is an alpha helix. Let's make it up by these in sequence here. So it goes from number, 310, all the way to number 320 or so. You had a question? No? Okay, so if you look at number 310, this amino acid will be pointing straight up here. Number 311 is pointed over here. It's about 90 degrees away and a little more than 90, right? It doesn't quite look like it's horizontal here. It'd be straight out this way. The number 312 is not exactly underneath. It's a little more. So in fact, the angle between these is roughly 100 degrees. Right? So if I go 4 amino acids around, I'm over here at 314, right? This one's 400 degrees from where it started. So each one is about 100 degrees, which means there's a slight twist as we go around. Right? So if I say there's 100 degrees between every amino acid pointing out in the next, how many amino acids would it take to go completely around the axis of an alpha helix? It's a very simple math problem.
4.
So 4 would be 400 degrees. That's too far.
Can you repeat the question?
So if it takes 100 degrees of turning, but from one amino acid to the next, how many amino acids would it take to go completely around in this alpha helix? So completely around is...
3.5, is that even possible?
Completely around is how many degrees?
360.
360 degrees. And if each time I go from one amino acid to the next, it turns 100 degrees, how many would it take to go all the way around?
3.6.
3.6 amino acids, exactly. So if you look at any particular alpha helix and pick a point along the chain, say right here, right, and you look at where that point occurs after it turns all the way around. So here, we're going to pick this point where it's turning from our point of view here, right there. Where is it turned all the way around again? That's right here. That distance, right, has been spanned by 3.6 amino acids. It's not a whole number because it's not a divisor of 360. It's 100 degrees. So it takes 360 degrees to go all the way around, and that takes 3.6 amino acids in length to turn that. And the reason we want to remember that number, 3.6, is I say, if this thing turns around 5 times, how many amino acids are in that alpha helix? Well, it would be 5 times 3.6, right? It's not a whole number, right? That's how many amino acids it would take to turn around 5 times. That's why you need to remember that 3.6 number. Right? The other type of protein secondary structure is called a beta sheet, right? This is an alpha helix, plural is alpha helices, but we have a beta sheet now. So beta sheets are very similar to an alpha helix in that they're held together by the same thing, hydrogen bonding, exclusively. It's the only thing that held the alpha helix together. This is the only thing holding the beta sheet together. So a beta sheet is made up of strands, right? So a sheet is made up of multiple strands. So here's a strand of amino acids. How do I know if this strand is going left or right? So here's one strand, here's another strand. You see their hydrogen bonding between them, right? How do I know if this strand is going left or right?
Is it the same thing with the NCC?
It's exactly the same. So this strand here, pick a nitrogen, let's use the one in the center. Is it going left or right? So if I try going left, I get N, alpha carbon, carbonyl. That's the correct order. If I try going right, I get N, carbonyl, carbon. That's not right. It should be N, alpha carbon, carbonyl. N, alpha carbon, carbonyl. The R groups are not shown on here. They've been deleted to make this clear because they're not involved. Right? So this top strand is going from right to left. How about the bottom strand? Let's pick a nitrogen. Which way is it going?
Left.
It's going right.
Right.
It's going left to right, exactly, because it's N, alpha carbon, carbonyl. N, alpha carbon, carbonyl. So this one's going left to right. So these are going in opposite directions. It's also possible that it could both be going in the same direction, like this one up here, right? N, alpha carbon, carbonyl. And this one is N, alpha carbon, carbonyl. So these are both going to the right. We call that, if both of them are going to the right, you see there's still hydrogen bonding going on here, right? These are called parallel strands of a beta sheet, or a parallel beta sheet. If they're going in opposite directions, like this one or this example down below, they're called anti-parallel because they're going in opposite directions. And again, you have hydrogen bonding. Of the two, the anti-parallel type is more stable because you have more hydrogen bondings per unit length, and they're perpendicular to the chain, as you do with parallels. Right? There are fewer hydrogen bondings per length, and they're not quite like they're shown here. They're going to be at a slight angle. This one's drawn a little bit too far to the right. They should be lining up, right? So they're kind of at an angle up here. So the anti-parallel arrangement is a little more stable than the parallel one, but I'm not trying to discredit the parallel one. It's still a valid, stable arrangement, just not as stable as an anti-parallel. And then you can also get something called a mixed sheet, where if you have more than 2 strands, of course, maybe you have 8 or 9 strands together, lots of strands. And at some point, they're parallel to one another, the neighboring ones, and maybe one nearby is going in the other direction. And the next one may be going parallel again. So it could be a mixed sheet. So it's not necessarily all parallel or all anti-parallel. You could have what's also called a mixed sheet, right? But it's also true you can have them all going in the same direction, or you could have them all alternating, and that would be a truly pure anti-parallel arrangement. So but all 3 arrangements are possible. But like the alpha helix, it's held together exclusively by hydrogen bonding, and only among the backbone amines and carbonyls. Right? Never the R groups. Again, I didn't even draw them. They're sticking off here and here and here. right? And unlike the alpha helix, where we said the R groups are pointing radially outward, so if you look down the barrel of this, they're all pointing outward, and there was that 100-degree twist, right? And it was hydrogen bonding from N to N+4 down the chain. This one has no such numerical relationship. The hydrogen bonding partner for, let's say, this one, right, isn't so many bases down, so many amino acids down the chain from, right? There's no numerical relationship. This strand could go off and do all sorts of things before it comes back here. All right, so no numerical relationship in a beta sheet. But the way the R groups stick out is quite unique, right? Because these are forming strands, you can imagine if there were several of these, like 1, 2, 3, 4, 5, 8 strands all on this page, right? There would be two sides of this sheet, right? There would be the side facing you, coming out of the board, and there would be the side away from us, into the board, right? Because they're forming like a little sheet. Like imagine a sheet of paper, right? These are the strands forming the threads of that sheet. And there's a face towards us, and there's a face away. And that's where the R groups are going to be pointed. They're not going to be pointed straight up and down here, because remember, this is tetrahedral. They're going to be coming towards us or away from us. And every other one will be facing the opposite direction. So if this one's facing towards us, the next R group will be facing away, and then towards us, and then away. And it alternates back and forth. So every other amino acid in a beta strand of a beta sheet, no matter if it's parallel, anti-parallel, or mixed, it doesn't make any difference. Every other one along an individual strand alternates which face or which side of the beta sheet it's on. Okay. Any questions on alpha helices and beta sheets so far?
Can you just repeat that last thing you said? I was writing it down. It's just a lot of information.
So on a beta sheet, the R groups are coming off in two ways, either towards us or away from us, since this is a tetrahedral arrangement. And because of the amino acids all being one chiral type, right, they're all going to be L amino acids, right, then they're going to come off towards us and then away, towards us and then away. It alternates back and forth along any one strand of a beta sheet.
Thank you.
Okay, so let's zoom out a little bit and see a little more of the protein. So we've been focusing mainly on the primary and secondary structures. Let's zoom out of our microscope and look at the protein on a larger scale. So we can see the whole protein now, not just an individual alpha helix or individual strands of beta sheets. We've zoomed out a little bit. And we're going to call this the tertiary structure. So we're looking at a larger view of the whole protein. And what tertiary means, it's often abbreviated as the 3 with the little degree symbol, called tertiary. Right? It's the overall 3-dimensional shape of the protein. Helps you remember tertiary. Right? So it's as if you can see the entire protein. Well, what shape does it take? Here's an example here. We could say this one is mostly spherical. Right? It's kind of an oblong pieces here and there. There's a couple alpha helices. There's some beta strands in the center that are curving on each other. But overall, it's kind of a globular shape. Right? So we can call it a globular protein. Right? And that's very common for soluble proteins. They take on a globular or round shape. It could also look like a soccer ball. It can look like a basketball. You can say it looks like a banana. Or this one looks like your uncle's toupee. Or you can be quite verbose in your language about tertiary structure, whatever it takes to describe it. Right? But then you can also say that it has about 50-50 alpha helical and beta sheet components. Right? Because these are smaller components of a larger structure. So this one's about 50-50 alpha helix, beta sheet, and there's some loops and turns here and there. Right? So you can also describe it as completely alpha helical. Right? That would be a good description of a tertiary structure, not this one, of course. But if it were nothing but alpha helices, then you can say the tertiary structure is a long thread or a bunch of alpha helices put together. But because you're describing the entire protein, that's the tertiary structure. Right? If I were to look at any individual alpha helix, that's secondary structure. That's a smaller, more focused view. So what held primary and secondary together exclusively? The only interaction that held them together.
Peptide bond?
Hydrogen bonding.
So both of you are right. So for primary structure, it was held together exclusively by the peptide bond. And in secondary structure, it was held exclusively by hydrogen bonding. They had one bond for each of those. For tertiary structure, we've got all of the above, whatever's possible can hold it together. You can have disulfide bonds, right? Remember we saw this with our our molecule earlier with our oxytocin and with the insulin molecule, right? We have, or vasopressin I showed you, right? We have a disulfide bond. So this can only occur between a pair of cysteines. No other amino acids can do it. No, methionine cannot. It must be two SHs that can interact, lose their H's, and become a disulfide. We can have electrostatics, right? These are salt bridges, they're also called. This is an ionic bond, right, between a positively charged and a negatively charged R group generally. So the R groups are certainly getting involved now. We can have hydrogen bonding all over the place between R groups, between backbones, between R groups and backbones, right? So all possibilities here. We also could have a lot of the non-polar R groups interacting with each other. These are not bona fide bonds. We just call them hydrophobic interactions. You may hear it as London dispersion or hydrophobic interactions or something like that. But they're not bona fide real bonds, and they're fairly weak. But in large numbers, they can be quite helpful in making the protein fold. Generally, the cores of soluble proteins are all hydrophobic in nature, so that they stay away from the surface and near the water, and it helps the protein hold its shape. So for a tertiary structure, it could be held together by any and all of these things, right? Not just one type. So this is the most diverse of the group. If you can think of an interaction, not the peptide bond, of course, but if you can think of any other type of interaction, even hydrogen bonding, tertiary can use it to hold together. Right? Everybody understand what tertiary means? We've kind of zoomed out and see the whole structure. And it can be held by any of these types. Okay, so one last thing to zoom out is quaternary structure. Now you're thinking, wait a minute, we just zoomed out to see the whole protein. How can we zoom out even more and have a larger structure? This quaternary structure is only possible if there's more than one protein chain interacting with one another. So back here on tertiary, you had one chain from start to finish. Maybe it started here. It goes through this entire thing, whatever it might be, and ends somewhere. Right? So this is one long amino acid train of molecules. Right? They're all connected by peptide bonds to one another. We consider that one chain. Right? But if I have multiple chains that are not necessarily connected to each other covalently, but there's two proteins that folded up on their own, right? And then they come together, or they fold together, but they're not connected to each other covalently by the peptide bonds. It's two separate chains that have come together to form a functional unit. This is when you can have quaternary structure. So those proteins that do not have quaternary structure, we simply call them monomers. There's one protein, it's a monomer, one part. Remember, mono means one, mer means parts. So monomer means one part. And if they have quaternary structure, they will not be monomers, they will be multimers. They have more than one part. So it could be a dimer or a trimer or a tetramer, depending how many parts it has. Right? So here's an example on the right over here from a protein that I've worked on. It has 4 different subunits, or 4 subunits. 2 copies of one in yellow here, and 2 copies of one in purple. Right? So there's 2 different chains here, and each appears twice in the structure. So overall, this would have a 4 subunit structure. Right? There's 2 of one type, we'll call the alpha chain. Maybe this one here in yellow or gold is called the alpha chain. And the second one we'll call the beta chain. It doesn't matter what you label them. We generally use the Greek alphabet to list them as alpha and beta, but which one's which really doesn't really matter. But in this case, they've labeled this one alpha and this one beta. So there are 2 copies of the alpha and 2 copies of the beta. Overall, there are 4 pieces of this structural unit. Right? So this whole thing has 4 pieces, so it's called a tetramer. Right? And this protein does have quaternary structure. In fact, it's a tetramer. Right? That's what its quaternary structure would be. But we have to be a little more specific in describing it, because there's lots of ways to make tetramers. You could have 4 copies of the same thing there. You could have 2 copies of one and 2 copies of another, as shown here. You could have 4 completely different things that come together. You could have 2 copies of one and a separate copy of a second one and another copy of a third type, and that would also be a tetramer. So there's many ways to come to a tetramer, so we have to be a little more specific. This one we'll call an alpha 2, beta 2 tetramer. Right? And more specifically, alpha 2, beta 2 heterotetramer. That means not all the components are the same. Right? Let me give you another example on the next slide in just a minute. A few examples. But what holds quaternary structure together? Well, everything that could hold tertiary structure together except covalent bonds. Right? So we're not going to consider disulfide bonds between these. Right? So if it's a tertiary structure, it can have these holding it together. But if we're looking at separate subunits, we'll discard the disulfide bond for here and look at all the other types that can hold it together. So what could hold tertiary together? Pretty much anything. Right? Not peptide bonds, of course, because that's in the primary. But what could hold quaternary structure together? Everything that we listed for tertiary except covalent bonds, so no disulfide bonds. Okay. So let's look at some examples of how to put this thing together. Just to review, primary structure is the amino acid sequence, start to finish. So in this structure at the top, it would begin here with the amino group. I would read these amino acids off, whatever their names might be, right, until I get all the way to the end here to the carboxyl group. Right, that's its amino acid sequence, that's the primary structure, and it's held together exclusively by peptide bonds. It tells me nothing about how this protein folds up. It just gives me the list of amino acids from one end to the other. That's all it does. Right, what can I get from primary structure? Could I get the mass of the protein from it? Sure, because I know all the parts. Could I tell you what amino acids are on the surface based on the primary structure? Absolutely not, because I don't know how it folds on itself. Could I tell you how many lysines are in the structure? Yes, because I have the sequence. Could I tell you if one of those lysines is bonding to a carboxyl group somewhere else in the protein? No, because I don't know how it's folded on itself. So lots of questions you can answer by primary structure, and many you can't. Secondary structure, these are alpha helices and beta sheets, and they're held together exclusively by hydrogen bonding. There was some numerical relationship for the alpha helix, right? There was not for the beta sheets. Tertiary structure. What does it look like if you zoom out and see the entire protein? Maybe it has this fish shape, right? Or maybe it looks like a boat. Or maybe it's mostly alpha helical with a beta sheet strand in the center. Or a beta sheet twist in the center. You can use lots of words to describe tertiary structure, but it's the entire protein. Right? That's all it means. It's the overall three-dimensional shape. What holds it together? Pretty much anything you could imagine, except peptide bonds. Those are holding primary together. Any other type of bonding pattern you'd like to describe could hold tertiary structure together. Quaternary structure. If there are multiple chains coming together, then you can have quaternary structure. In this case, we have several chains here, colored different colors for you here. So maybe 1, 2, 3, 4 chains coming together, 2 of one type and maybe 2 of a different type coming together, or maybe these are different from each other. But all together, those are quaternary structure because multiple subunits or multiple chains have come together. Right, so let's look at some examples of those quaternary structures. Right, so here's some examples. I have 2 total peptides, and they're identical to each other. What would you call that? It does have quaternary structure because this is greater than 1, but it has 2, so what would you call it if it has 2 total peptides?
Dimer?
It'd be a dimer. Excellent. And they're both the same, so be more specific about dimer. How would I indicate they're the same? What's the prefix for same?
Homodimer?
It's a homodimer, exactly. Right, so we call that one a homodimer. You don't have to put the alpha 2 here. It's a little redundant, so I won't show you this. I'm just letting you know it'd be like this one. I've colored them different colors, but you can see they're identical to each other. They're just facing one another, two copies of the same chain. Right, so it's called a homodimer. You don't need the alpha 2 because it's kind of implied here that they're the same. Right? What if you had 2 total, not 4 here, but 2 total peptides and they were not the same? What would you call that?
Hetero?
Hetero what?
Of 4 of them?
No, there were just 2 total, and they're not the same.
Heterodimer.
It'd be a heterodimer. Again, I don't have to tell you it's an alpha and a beta because dimer kind of implies there are 2, and if you say hetero, I know they're not the same. Right? So a heterodimer or a homodimer don't need prefixes. All homo types don't need prefixes like homotetramer, homooctamer, whatever, you know, it's all the same type. But heterodimer also doesn't need a prefix because you know it's one and then a different type. It'd be an alpha and a beta.
The one that you showed us on the previous slide, the one that you said you did, with your name on it,
Yep.
is that a hetero? Would that be a heterotetra?
That's the one I'm about to show you next right here as well. It's 4 total peptides, 2 of each type, right? Same exact one, but different protein, just same arrangement. It's a heterotetramer because there are 4 total strands there. Right? 4 total chains. 2 of one type, 2 of another. It's like writing a chemical formula here. It's 2 of alphas and 2 of betas in this thing. 2 of the green ones and 2 of the blue ones. They're not the same as each other, but the blues are the same and the greens are the same. Much like the ones I showed you on the previous slide. Okay, so this would be called an alpha 2, beta 2. Which one you call alpha, which one you call beta doesn't really matter, right? But you got to name them separately. Heterotetramer. An easy way to figure out it's hetero is there aren't all one type, right? That's pretty easy. Tetra means add up all the subscripts, right? There's 2 of this one and 2 of this one, right? How about 3 total peptides? 2 of them are the same, and the third one is not like the others. What would you call this? So 3 total peptides. What would you call that first?
Tri.
A trimer, right? 3 total peptides would be a trimer. Peptide is just a name for a chain, right? 2 of them are the same and 1 is not. So we have to describe it. It's not all the same. So it's a heterotrimer because not all of them are the same. They don't all have to be different. They just can't all be the same. Right? So 3 total peptides, not all the same. Heterotrimer. And then how do I tell that 2 of one type and 1 of another?
Alpha, beta.
Right, alpha 2 beta. So 2 of this one and 1 of that one. Right, and here's an example of that. 2 of these are the same. It's kind of hard to tell which 2 are the same, but 1 of them is different from the others. Right, but 2 of these are the same and 1 is different, so it's a heterotrimer. Another type of heterotrimer would be if all 3 are different from each other, and you would list that as alpha, beta, gamma heterotrimer. Again, just add up all the subscripts, 2, and there's an understood 1 here, much like your chemical formulas. And then lastly, I have 6 total peptides present, all of one type. What would you call that? So 6 total peptides would be called a what?
Hexamer.
A hexamer, right? Like trimer or dimer or tetramer, but hexamer. And they're all of the same type, so homohexamer, exactly. I don't need any prefix in front of it, right? I don't need the alpha 6 because it says homo. You never need a prefix for that because you know they're all the same. It'll always say the same thing with whatever number goes there. So you don't really need that. I'm just showing you what it would look like. And here's 6 copies of the same protein all interacting with each other to form this homohexamer. And there's lots of other options. These are just some examples. Look over the practice test and you'll see some other examples. But all of these are describing quaternary structure. If I say there's a single peptide chain all by itself, we consider it to not have quaternary structure, and we just call it a monomer. That's it. There's nothing attached to it. And we consider the monomers to not have quaternary structure.
I have a question.
Uh-huh.
If the picker didn't specify for the one about 3 total peptides, what if it was like alpha 1, beta 2? Is that also, couldn't that also work?
Right. If you could write alpha, beta 2, right, the one would be understood. That would be the same thing, a heterotrimer. Exactly.
But if we were to have to identify it, it would be stated in the picture, we would know what to put for the subscripts. Or... Right.
It's really a whatever you like. Whatever subunit you call alpha and a different one you would call beta. If there's 2 of one and 1 of another, you could write alpha 2 beta or alpha beta 2. Both of those are equivalent. Just which one you call alpha, which one you call beta is up to you. Generally, once it's been established in the literature which one's called which, we kind of stick to it. Right? But for a new protein, you're free to call any one you want, any letter you like. The common trend is to give the more numerous one the first letter, or the larger one the first letter. But you're free to choose any way you'd like to do it. But it would be a poor idea to keep switching in your paper which one you call alpha and beta, right? You want to stick with it.
Okay, thank you.
All right, and then the last thing, you may have heard this before in nutrition or in some other classes where they described what foods you should eat and not eat and disease processes for missing foods. And at the top here we have our what we call the essential amino acids and non-essential amino acids. And almost everyone agrees on 19 of the 20 amino acids. Right? So here's my 20 amino acids listed. right? These over here, these 9 are listed as essential, and everyone agrees that these are essential. And by essential, we mean that humans lack the enzymatic machinery to synthesize these. Right? So we can't make these amino acids. We have to get them in our diet. That's why they're called essential. We must get them in our food. And over here, we have 11 listed, and the top 10 here, not tyrosine, but the ones above it, everyone agrees that they're non-essential. We have the molecular machinery to make these 10 amino acids. Everyone disagrees on tyrosine. And it all depends on your definition. And I agree with both camps. It all depends on your definition of essential. If your definition is, can you make this amino acid from any other raw material in the body, then tyrosine belongs in the non-essential category. Because we can make tyrosine, we have the enzyme to make tyrosine from phenylalanine. It's just adding an OH group to it, right? So we can make tyrosine from another thing that we have in the body, phenylalanine. However, if I don't have phenylalanine in my diet, I can't make tyrosine. So that might be a reason to put it over here in the essential category. So if you can't make it from scratch, right, or de novo, which means from nothing, right? If you can't make it from some simple molecules like glucose, then tyrosine belongs over here in the essential category because you can't make it from other things, not the other amino acids. So it all depends on your definition of what can you or can you not make it from. Right? If those are your reasons for putting it in the essential or non-essential categories, I totally agree with your reason. Right? So if you put it over here, you're saying you can make these things from other things in the body, like phenylalanine can be made into tyrosine. I agree with you. If you put it over here and say, I put it in this column because I can't make it from scratch or from raw materials like glucose, then I agree with that as well. If you put tyrosine in this column because you wanted 2 nice columns of 10 without an explanation, I disagree with that reason. That's not a valid reason for putting it in that column. And a lot of textbooks will do that and not tell you why tyrosine was put in this column. Right? So depending what your rationale is, I agree with putting it in either column as long as you have the appropriate rationale. Right? So what's shown here is we can make these from scratch, right, or from some raw material, excluding tyrosine, and we cannot make these on the essential column. So a lot of times you'll see these as fortified in foods, right? Especially these branched ones like leucine, isoleucine, right? And other branched ones over here like threonine and valine. But you'll see these listed on energy drinks and often on foods where they're trying to get a high protein content. And you need these more than you need these because we can make these from scratch in our diet, or from other things we get in our diet. Right, now this little guy here, right? Does he look healthy to you?
No.
What appears to be wrong?
He's bloated.
Right, so his abdomen is distended, right? So do you think he's malnourished in any way?
Yeah.
Yeah, I mean, from a very crude view, if you have no other understanding about this at all, it might look like he's got a full belly, right? He's well fed, but that's not true at all. He's getting lots of food, but he's not getting one major thing in his diet. His diet is lacking in one major component, and that's protein, right? So he's getting lots of carbohydrates and maybe lipids, but he's not getting enough protein in his diet, right? So he's got a distended abdomen. Why is that the case? Well, think about it. If you eat a lot of protein, right, and then, or in a normal diet, you're getting a lot of protein in your diet, no matter what foods you're generally eating, unless you're suffering from a malnutrition like this. When you start digesting those proteins, they're pulled apart into individual amino acids, right, and they're absorbed in the gut, and then they're sent through the bloodstream. Most of them are fairly soluble, and so there's lots of amino acids in the blood, right? And so your blood is full of things like sugars and amino acids and other proteins and other components and other salts. There's lots of stuff in the blood, right? And then compare the blood components to the tissues, right? The interstitial fluid and the space between cells and the internal spaces of organs. Compare that fluid to the blood. The blood will have roughly the same amount of components as the parts in the interstitial fluid. But if you're lacking protein in your diet, a condition called kwashiorkor here, then you'll have a low concentration of amino acids in your blood because you're not getting many in your diet, right? It's not being absorbed very well in your stomach or through the intestines, and the amount of amino acids in the blood will drop drastically. That's what's happening with this little guy. So the amino acid concentration in the blood is what compared to the interstitial fluid? Use some terms you've learned. The blood would be considered what compared to the interstitial fluid? It ends in tonic.
Hypotonic.
Hypotonic, right? It's got fewer things dissolved in it compared to the interstitial fluid, right? It's hypotonic. Or if you consider the interstitial fluid compared to the blood, that would be considered a hypertonic compared to the blood, right? You can say it either way. What happens in those scenarios?
It leaves the cell and then goes to the stomach.
What's the pronoun it referring to?
The water.
So water will move between the two areas, right? So the other things are hard to move between membranes, like amino acids and the salts and stuff are hard to move across. Water is very easy to move across, so it's going to move first. So in this case, since the blood is hypotonic and the interstitial fluid is hypertonic, the water is going to flow to the interstitial fluid. So it leaves the bloodstream and goes into the interstitial fluids, which causes the edema or the swelling in those tissues. And the most likely place for that to occur is in an area where you can accumulate a lot of fluid, and that's going to be in the abdomen, right? So that's why he has this protruding abdomen or this edema. So if you had someone like this and you run across this child while you're out jogging or whatever, you see someone like this, they need protein in their diet. So what would you do? I always get the same answer to this, and it's always wrong, but say it anyway.
People would say give protein.
Yeah, people would say give him a protein bar. That's a very dangerous thing to do, because what would happen if you gave him a large bolus of protein, he eats it, it gets into the bloodstream, and now there's a ton of protein, not literally, but lots of protein broken down into amino acids in the blood. Right? The blood is already at a lower volume. You've introduced a lot of amino acids in there. Where's all this fluid going to go?
Back into the blood vessels?
Back into the blood. So what's going to happen to the volume of blood?
His blood pressure is going to go up.
The volume's going to go up, which increases the blood pressure, the blood volume, and he might have a heart attack, right? Or he might leak out of a vessel somewhere, an internal bleeding, or he might hemorrhage in the brain. So giving him a large amount of protein all at once is a very bad idea. So you give him, assuming he's not allergic to peanuts, give him a peanut. Right? Give him a small amount of protein. And then come back in 20 minutes, I'll give you 2 peanuts. Come back in a half hour, I'll give you 4 or 5 more. So you slowly build up the amount of protein in his diet over time, and then you can cure this swelling. Let the fluid go back into the bloodstream and fill out the body in a controlled manner, not suddenly and dilute the blood of electrolytes and cause the higher blood pressure.